Hello friends, Ace Botany, next episode. In this episode, we'll be discussing last part of the principles of inheritance and variation, 8th one. So we can proceed to the respective episode. So I will share the screen, it will be convenient for you. I think screen came. So we can proceed. So what all things we'll be learning in this episode? Mainly 3 items, that is pedigree analysis, Mendelian disorders, chromosomal disorders, and after that, as usual, 5 theory question, 5 NEET model question with the key. So we can enter into the area. So first is pedigree analysis. So we should know what is meant by the term pedigree. See, pedigree is a diagram or family tree representation by symbols of certain genetical traits or events for 2 or more generations. So it is originally a representation, diagrammatic representation with the symbols. Now, when we deal this pedigree analysis, so 2 basic terms, that is proband or propositus. What is meant by that 2 terms? They are synonyms, proband or propositus. It means starter of a pedigree analysis, from where that pedigree should be analyzed. That individual is called proband or propositus. I think up to that it is clear. Now we can find symbols on the left-hand side. This is purely NCERT, usual area for question, area as well as NEET exam. So male, female, then sex unspecified, affected individual, that is with a shading, male with a female, single line, that is mating, and male with a female, 2 lines. That is the most predominant question from this area. Mating between relatives, which is otherwise called consanguineous mating. Then a tree represented parents with the offsprings. Then parents, where we can find affected child, shaded. And last one, 5 unaffected offspring with a number. So these are 5 common direct NCERT symbols. Now when we come to this side, in human genetics, what is advantage for pedigree? Different views are there. One view is, in human genetics, pedigree study provides a strong tool which is used to trace the inheritance of specific trait or abnormality or disease. That is the advantage in human genetics. I told you different views are there. And specific symbols we represented here. After that, some more symbols are there which came for some other previous exam. These all are not in direct NCERT, but still. So these 2 stands for death, male, female, etc. This is abortion or stillbirth. These 2 terms, it is concerned with the proband or propositus, where if it is male, it is called propositi. If it is a female, circle you can find shaded, proposita. And these 2 stands for twins. This is monozygotic twins. That is, sex will be same. That is why this has been shown, indicating it is monozygotic. This is dizygotic twins, where that bar is not shown here. So sexes can be different, sexes can be same. Now a pedigree tree has been shown here, where it belongs to NCERT. One is autosomal dominant trait. Example is myotonic dystrophy. Second is autosomal recessive trait. Example is sickle cell anemia. Many type of representation is there. Autosomal dominant, autosomal recessive, sex-linked dominant, sex-linked recessive, crisscross, Y-linked. That way different representations are there. That is regarding the first page, pedigree analysis. Now we can proceed to second page, that is dealing with the genetic disorders. Genetic disorders generally grouped into 2. One, Mendelian disorder, other is chromosomal disorders. So first we'll start with the Mendelian disorders and what are the features of Mendelian disorders. How it differs from chromosomal disorders. See, in Mendelian disorders, it is gene related. Usually mutations in single gene. That is first idea. Second one, inheritance of the offspring in a Mendelian principle, based on Mendelian principle. Third, pattern can be traced in a family by pedigree analysis. That are the features of Mendelian disorders theory NEET exam question. Two types will be there as usual, dominant and recessive. Similarly, autosomal section, etc. So this is a table showing that genetic disorders, Mendelian disorders, where we can find recessive and dominant. It's a table only. It is not seen in present NCERT. It is old NCERT, previous edition box. How we have to learn it is name of the disease, condition, dominant or recessive, autosomal or sex-linked. If it is autosomal, is it chromosome number, then symptom as well as effect. After this table, 3 diseases I shown there. Thalassemia presently in NCERT still, then myotonic dystrophy, DMD. DMD is largest human gene. It is recessive, X-linked old age. It codes for a protein dystrophin connected with the muscle. Myotonic dystrophy, that is connected with the muscle, etc. Still, it is dominant. And it is autosomal, long arm, chromosome number 19 in young age. So there is a difference, or there are differences between myotonic dystrophy and DMD. Cataract, heart, breathing. Again, here also muscles, etc. Thalassemia will be learning in detailed manner. So myotonic dystrophy and DMD. Others are theoretical. If you can by heart, it is good. Usually 1 or 2 questions we may be getting for NEET and theory exam. We can proceed to next page. Now the diseases, conditions. First is colorblind or color blindness. Wilson in 1911. Again, certain other scientist name also proposed. And this condition, colorblindness, also called Daltonism, otherwise called red-green colorblindness, tested by Ishihara test or chart. This is a famous chart, reading the number with the different colors, etc. If you are blind for a color, you will not be able to read that number. Now in driving test, etc., this is becoming important because signal red and green mainly. Now colorblindness is sex-linked, X chromosome, recessive, and since it is X-connected, quick expression in males because males have only one X. Now a cross shown, colorblind man, normal woman. So from colorblind man, 2 types of gamete. Here you can find woman, normal woman, only 1 type, coupling, combining, and you will be getting this checkerboard. You can find no girls or females will be disease. They are carriers. Males all will be normal. Now just another combination. That is, here we can find another one, normal man and colorblind woman. So normal man means two type of gamete. Colorblind woman means that is recessive. So both should contain, both X should contain C. So only 1 type of gamete coupling, and we'll be getting carrier girl. Again, no disease, and we can find colorblind man or boy. Since female side, 1 gamete will be, or all the gamete will be XC. So nutshell, here 2 carrier girl, 2 normal boys. Here 2 carrier girls, and we can find 2 color blind boys. I think idea clear, simple. Now we can proceed to next one. Colorblindness is X-linked recessive trait. We can proceed to next one, that is hemophilia. Scientist is John Otto, initial worker. Again, some other names are there. 1803. It is commonly called royal disease or bleeder's disease. X-linked recessive, just like previous case. To get the disease in males, chance is more due to XY. Since X has got no complement, that is only Y. So immediate effect will be there, what all characters appears in X in males. Female normally act as a carrier, but female also can get it, but normally act as a carrier due to XX condition. It is recessive sex-linked gene, namely small h. 3 types of hemophilia, hemophilia A, B, C. Hemophilia A is called classical hemophilia. Hemophilia B is called Christmas disease. Hemophilia C is called Rosenthal syndrome. Hemophilia A, that is classical hemophilia. It is due to fact number 8, which is otherwise called anti-hemophilic factor or anti-hemophilic globin. Hemophilia B, Christmas disease, fact number 9, it is called plasma thromboplastin combinant. And hemophilia C, that is not in our syllabus still, Rosenthal syndrome, fact number 11, it is plasma thromboplastin antecedent. Hemophilia is connected with the deficiencies in blood clotting, no blood clotting delay, etc. Possibility of a female become hemophilic, chance is comparatively less because 2X chromosome should bear that gene. father should be hemophilic and mother must be at least carrier. Then there is a chance for the daughter to get the disease. And it is connected with the family of Queen Victoria, royal families. So that is why there is a name for this disease, royal disease. A belief of many members, either carrier or disease due to hemophilia B gene of Queen Victoria. More chance for a female hemophilic patient to die before birth. Inheritance is just like color blindness. That is regarding hemophilia, and interestingly, that hemophilia day. Normally it is April 17th, World Hemophilic Day. Then comes another disease called sickle cell anemia. Name is due to sickle shape RBC cell caused by the disease condition. It is autosomal recessive condition. Chromosome number is 11. Offspring will get the disease when both parents are minimum heterozygous condition, since it is homozygous recessive condition. It is autosomal, not sex-linked. 3 combinations are possible. One is normal, one is carrier, one is disease. So normal is HBA, normal homozygous condition. Carrier is HBA, and disease condition is HBS. And patient may even die in that HBS condition. Now, these are the diagrams of NCERT. It's a common question, diagramized question. And a part of it I enlarged here. There we can find normal HBA gene. This is sickle cell condition. So one by one we can proceed. Normal case, here you can find mRNA, it is GAG. Disease condition, it is GOG. These are the peptide chain. Polypeptide chain, normal, disease, or HBS. Now, this is the essence of it, at least try to remember it. Sickle cell anemia type of mutation is transversion. The chain is beta chain. Codon is 6th codon, assembly of 3 bases, 6th one, say 1, 2, 3, 4, 5, 6, 6th codon. Each codon 3 bases, so 6th codon means we'll be getting 16, 17, and 18. Up to 5, 15, 16, 17, 18. Middle base is changed, so base number is 17. Shape of RBC changes from biconcave disc to elongated due to oxygen deficiency, etc. disease condition. Carrying will be affected for oxygen. Amino acid change, that is important. Glutamic acid to valine. mRNA change. Glutamic acid is GAG. Valine is GUG. That you can find here. Here you can find. So that part is very important for sickle cell anemia and usual theory entrance question. Hoping that it is clear to all of you. Now comes next disease condition, phenylketonuria. Nuria indirectly means a condition or an event. Meaning will be variable, usually coming as a suffix. Phenylketonuria short form is PKU. Another one is called alkaptonuria, short form is AKU. They are commonly inborn errors of metabolism. Inborn errors of metabolism, IEM. It is an autosomal recessive disorder. Here you can find chromosome number is 12. An enzyme deficient in this phenylketonuria is a liver enzyme phenylalanine hydroxylase. And due to its absence, amino acid phenylalanine cannot be converted into tyrosine. Phenylalanine will accumulate in blood and brain, get converted into phenylpyruvic acid and other derivatives. Accumulation in brain leads to mental retardation. And some of it may be excreted through urine because kidney poorly absorbs it. And remember, a term we studied earlier, pleiotropism, single gene, many effect. Alkaptonuria, that is not in our syllabus, that is typically AKU, is also called as black urine disease connected with alkaptones or homogenetic acid. That is regarding phenylketonuria. Now comes next condition, disease condition, it is thalassemia. Thalassemia have different meaning. Here it means sea or ocean. Amia, AEME, amia connected with the blood, health, condition, etc. Here sea is referring to Mediterranean Sea. It is an autosomal recessive hemoglobin-based disorder. 2 chromosomes will be involved, chromosome number 11 and 16. It may be connected with a group of disorders connected with the synthesis of globin polypeptide in RBC, mainly alpha and beta globin. If one globin is less or deficient, then other globin will be excess. Symptoms include damage of RBC, cell lysis, anemia, etc. Now comes to thalassemia, alpha thalassemia, beta thalassemia. In alpha thalassemia, two genes involved, HBA1, HBA2, chromosome number 16P. Part is somewhat a little bit deleted. That is chromosome number 16P. Part will be deleted, part of it. Then P means short arm. Total 4 alleles will be there. 2 gene, 4 alleles. And decreased alpha globin production, excess beta globin in adults, excess gamma chains in newborns, excess beta chain will produce many abnormalities connected with the hemoglobin, etc. That is alpha thalassemia. Connected with the chromosome number 16P, part will be deleted, a portion of it. Now, beta thalassemia, only one gene. It is HBB on chromosome number 11, 2 alleles. Now, thalassemia major, thalassemia intermediate are available. Delta thalassemia also seen linked with the chromosome number 11. And these two statements are very common for exam. Thalassemia is a quantitative problem because more alleles. Synthesizing too few globin molecule, 2 TOO. While sickle cell anemia is qualitative problem, synthesizing incorrectly functioning globin. So there is a difference between thalassemia and sickle cell anemia. Thalassemia is quantitative, sickle cell anemia is qualitative. Common for exam. Now we have to proceed to next area, chromosomal disorders. So chromosomal disorders are caused due to absence or excess or abnormal arrangement of one or more chromosome. That is the definition given in NCERT page number 75, March 24 reprinted issue. Typically aneuploids and polyploid, we already studied that still. Now under which chromosomal disorder, first one we have to learn is Down syndrome. Common name is Mongolism or Mongolian idiocy. First reported by Langdon Down in 1866. And reason is due to trisomy in 21st chromosome, autosomal abnormality. Then it is due to how the trisomy comes non-disjunction in 21st chromosome. As a result, both the chromosome will be passing into single egg, where chromosome number become 24, resulting in 47 chromosome inside. So it is hyper. And this is the diagram given in NCERT, an individual with the typical Down syndrome. This feature is very common for exam. Commonly, that patients will be physical, psychomotor, mental retardation will be there. And this is the famous ideogram or karyotype for Down syndrome where 21st chromosome trisomy we can observe. I think idea clear. Now we will proceed to next syndrome concerned with chromosomal disorders, Klinefelter syndrome. It is X-linked allosomal abnormality connected with X chromosome. Here it is normally hyperploidy, 47, above 46, normally 47. How this 47 or higher level comes? It may arise due to C combination you can find. Either here you can find 2X in gamete, male is normal, so 44 plus XXY. Or female is normal, male is abnormal, again 44 plus XXY. This is how the Klinefelter syndrome is caused, hyperploidy. And what will be the nature of the individual? Again, first diagram, it is NCERT based. Individuals will be normally male, sterile. Generally mutation means it will be sterile, like hybrids. Appears more like a male with a few female characters like sound, development of breast. This is common for exam. The condition is called gynecomastia. Features given for Klinefelter syndrome. Now comes the last syndrome for us. It is Turner syndrome. Again, allosomal connected with the sex chromosome X. It is hypoploidy, lower than 46. It is monosomy 2n minus 1. How condition can be caused? 22 plus 0, egg is 0, plus normal 22 plus X, or 22 plus 0 sperm, plus 22 plus X. So total will be 45. Why you cannot find here? So normally individuals will be female. They will be sterile with a rudimentary sex organ. Male Turner syndrome is theoretically 44A plus Y0. Theoretically, it will be 44A plus Y0. After that, some terms or events came for different exams. So first is trisomies. 3 trisomies, only one will be learning. Down syndrome, 21st trisomy. After that, Edward syndrome, 18th chromosome trisomy. Then Patavo syndrome, 13th chromosome trisomy. So 3 trisomies. Down is within syllabus. Other 2 you can remember. If any question comes, you can answer. Now 2 famous deletions. 5th chromosome, part of short arm, 5P. P means petit. Petit in French means short. So 5th chromosome, part of short arm, 5P, deleted. It is causing cry-do-shat syndrome, cat cry. Similarly, 11th chromosome, part of long arm deletion, 11Q. It is causing Jacobson syndrome. Now, next item, XYY is called Jacobs syndrome. Jacobs syndrome connected with the antisocial tendency, etc. So these 3, again, important for exam. Yeah, that is why I just mentioned 2 deletion and 1 XYY condition. Deletions, Cry-do-shat, Jacobson syndrome. Then Jacobs syndrome is XYY. I think idea clear. Now, after that, 5 theory question, 5 NEET model question with the key. So what all things we learn in this episode? Last part of principles of inheritance and variation, 3 items, pedigree analysis, Mendelian disorders, chromosomal disorders. In pedigree analysis, symbols, 2 family trees, then genetic disorders. Genetic disorders, you can find what is genetic disorders, then Mendelian disorders, then a table. After that, 3 other diseases. Then each one, color blindness, name, features, chromosomes, sex-linked, etc. Then hemophilia, ABAC, and how that will be inherited, just like previous case. Then sickle cell anemia, that is the next one. Autosomal. Then what are the conditions? What type of mutation? Which chain, which codon, which base, which amino acid, etc. Then phenylketonuria, that is the next one. PKU, AKU, autosomal, recessive, chromosome number 12. Now, enzyme, amino acid conversion, then accumulation, pleiotropism, alkaptonuria, AKU, black urine disease, then homogenetic acid. Then comes thalassemia, ocean, then thalassemia, alpha thalassemia, beta thalassemia, that is to within syllabus 11, 16. Then globin production, 1 is deficient, that will be excess. Then alpha thalassemia, beta thalassemia, genes, alleles, chromosome. Then delta thalassemia also available. After that, in alpha thalassemia, you can find chromosome 16P, short arm. Part will be deleted, a part of it will be deleted. Then quantitative and qualitative thalassemia, sickle cell anemia, important for exam. Then chromosomal disorders, that is Down syndrome, 21st trisomy, how it has been caused. Then after that, connected with the sex, Klinefelter syndrome, again hyper, 47, how it is caused, what are the features. Then Turner, it is hypo, 46 minus 1, again what are the features. It normally comes in female, male Turner syndrome, normally theoretically possible, but may not occur. Trisomies, Down, Edward Patavo, deletion 5th, 11th, then XYY, their names, etc. And after that, usually 5 theory, 5 NEET model question with the key. Hoping that you are able to follow this episode. Exam point of view, comparatively more important theory entrance. And if you like this episode, please subscribe all, like it, share it, comment on it. We can meet in another episode. So take care. Thank you.